Thank you very much, and I appreciate the opportunity to share some ideas with you all. What I'm going to talk about today is this notion of an agential material for bioengineering. And so we will talk about bioelectric mechanisms and things like this, but the important part of this talk is not so much the fact of this biophysical mechanism, but what it tells us about information processing and in particular diverse intelligence that goes all the way down to the biological material. So if you want to download any of the papers, the data sets, the software, all the things I talk about are here at this website. And here are the main points that I would like to transmit today. I'm going to start off with discussing the context that most problems in biomedicine and bioengineering really are fundamentally about the control of shape, and in particular about the collective decision-making among cells. And I will argue that this is not going to be solved by hardware-based approaches such as genomics and molecular biology, but actually is going to require a lot of thinking that comes from the cognitive sciences. Because biology uses a multi-scale competency architecture. In other words, we consist of nested problem-solving agents that work in all sorts of unconventional problem spaces. Engineers and also evolution exploits this architecture, and it's this bioelectrical interface that allows cells to behavior shape each other towards creating and maintaining complex anatomical form. We now have tools to read and write the pattern memories of the software layer of life, that layer that sits between the genome and the anatomy. And there are numerous applications in birth defects, regeneration, cancer, and synthetic bioengineering. And in particular, kind of the main message of today is that when you are engineering with agential materials, you can use tools from the behavioral and cognitive sciences to exploit the collective intelligence of cells as it operates in these problem spaces. So let's just work backwards from what I think is the end game of biomedicine. Someday, you will be able to sit down in front of a computer and draw the animal, plant, organ, biobot, any kind of living system that you want, draw it at the level of anatomy, not at the molecular biological level. And what the system will do then is to compile your goal, your description, into a set of stimuli that would have to be given to cells to get them to build exactly this, in this case, a nice three-headed flatworm. Now, if we had this, all of these things would go away. Birth defects, traumatic injury, cancer, aging, degenerative disease, all of this would go away because we could tell cells to build and rebuild all the complex, healthy structures that we need. So how would we do this? And in particular, why don't we have anything like this? You know, molecular biology and genetics have been going very strong for decades. Why don't we have this? And I'm going to argue that here's what's happening here, is that this anatomical compiler, this thing that's going to allow complete control over growth and form, is nothing like a 3D printer. It is not about micromanaging the details as you would when you were engineering with a passive material such as Legos, where you're in charge of every single piece. It's actually a communications device, because we in biology are not dealing with active matter or even just computational matter. We are dealing with an agential material because we're made of competent parts with agendas and problem-solving capacities. And so the goal is to transmit the needs of the regenerative medicine worker or bioengineer to the collective intelligence of cells, so that that is what they build. So here's what we are made of. I mean, this is a single cell organism, and this happens to be a lacrimaria, which is a free living organism, but this just illustrates what individual cells can do. This thing has no brain, no nervous system, but you can see it's very competent at handling its tiny little agendas in physiological space and metabolic space and so on. And all of us started life this way. We all began life as basically an unfertilized oocyte, a little blob of chemistry and physics. And then eventually, through this incredible process of embryogenesis, we became one of these things, or even something like this. So this is René Descartes, a complex metacognitive human who will make comments about not being a machine and being, you know, something more than physics and chemistry. But this process is slow and gradual. So we all have this origin, and so that means we need to understand how this works. How did we get here from here? Now, I will point out that there is even intelligence below the cell level, meaning that it's not just that the individual cells are competent, but even below the cell level, there are pathways So like gene regulatory networks and other biochemical pathways that you may think of as mechanical, as deterministic, hardwired kinds of things. But in fact, if you pay attention, it turns out that they are capable of learning. They show about 6 different kinds of memory, including Pavlovian conditioning. Not neurons, not brains. You don't even need the cell. It's the molecular networks themselves that can learn. And in our lab, we are currently building devices to actually take advantage of the learning capacity of cells and do things like drug conditioning and other ways to control the molecular pathways, memories, and the expectations that they have and how they deal with future stimuli and so on. So here's our architecture. We are not just nested in a sort of structural sense. I mean, everybody knows, molecules, subcellular components, and organs and organisms and swarms and so on. But it's not just that. It's the fact that each one of these is itself a competent agent navigating things like anatomical space and transcriptional space and so on. And so there are these autonomous competencies at every stage. Now, we as humans are primed to recognize intelligence in 3-dimensional space, specifically medium-sized objects moving at medium speeds, navigating 3-dimensional space. We can look at that and we can say, okay, we observe some kind of problem-solving capacity. And we're actually not good at all at recognizing the same thing when it occurs in other spaces. For example, cells navigate the space of all possible gene expressions. So that's transcriptional space. That's a pretty high dimensional space. They navigate physiological spaces, and they navigate anatomical morphospace, the space of all possible anatomical configurations. This is the one we're going to talk the most about today. And I need to show you what I mean when I say they navigate the space, because you might have the idea that that morphogenesis is the result of a kind of mechanical working out of the rules of biochemistry. And while that's not exactly false, it's a very limiting picture that does not give access to the opportunities of regenerative medicine. And I'll show you what I mean. So let's first ask ourselves where anatomies come from. So this is a cross-section through a human torso. You can see all the different organs, the structures, everything is in the right place relative to each other and so on. Where is this pattern coming from? Well, because we all start life like this, so this is a set of embryonic blastomeres that are going to build this, we need to understand, well, where is this pattern specified? And you might think it's in the DNA. That's the most common thing people say, is it's in the genome. But we can read genomes now. And we know that this is not directly in the genome at all. What the genome specifies are proteins, the tiny molecular level hardware that every cell gets to have. But the final outcome is not directly readable from the genome any more than the shape of a termite mound or the shape of a spider web is readable from the genomes of these creatures. So we need to understand what is the software layer here. How do the cells know what to make? How do they know when to stop? If something is missing or damaged, how do we convince them to repair and rebuild? And as engineers, we would also like to know what else is possible. They build this, and what else can we get the exact same cells to build? And we want to focus a little bit on this genetic information. This is just one example. Here's a baby axolotl, and these juvenile axolotls have little legs. Here is a tadpole of a frog. They do not have legs at this stage. Now, in my group, we make something called a frogolotl, which is an early chimeric embryo that has some frog and some axolotl cells. We have the genomes. We have the axolotl genome. We have the frog genome. They've been sequenced. They've been read. Now, I ask you a simple question. Given this, can you tell me if a frogolotl is going to have legs or not? And if it is going to have legs, are those legs going to be made entirely of axolotl tissue, or some of the components will be frog cells? And the answer is there's no way to tell. We have no way of knowing from this genetic information what's actually going to happen here. So while we are, as a field, we're very good at manipulating molecules and cells, and so making these kind of pathways, who interacts with whom, and so on, we're quite good at this. We're really a very long way away from predictive and control of form and function. So if somebody's missing an arm, how do we convince the cells to regrow it, or to explain the form to begin with? We have a lot of difficulty with that still. And I'm going to argue that's because biomedicine has been largely focused at the hardware level of analysis. It's kind of like what computer science and information technology was like in the 1940s and 1950s. So here she is, she's reprogramming this computer. And in those days, you had to interact with the hardware. In order to reprogram it, you had to move wires around. Okay, and this is what's going on today. All the most exciting advances of molecular medicine, genome editing, pathway rewiring, protein engineering, all these things are down at the level of the hardware. But of course, nowadays, if I told you that in order to switch from Photoshop to Microsoft Word on your laptop, you have to get out your soldering iron and start rewiring, you would laugh. And why is that funny? Because we know what's happened with the field of computer science. It spawned the age of information technology by realizing that if your hardware is good enough, and I'm going to argue that biological hardware is definitely good enough, then you have access to something very powerful, which is reprogrammability and intelligence of the material. You don't have to rewire it to get new components. And so here we have to have a definition. I've already used this word a couple of times. I'll use it many more times. Here's what I mean by intelligence. It's basically William James's definition, which is the ability to reach the same goal by different means. Okay, notice it doesn't say anything about brains. It doesn't say anything about what kind of goals or what space, what problem space your goal is in. It's basically the ability to navigate some problem space to get your goal met despite various perturbations, barriers, novelty, and so on. And so this is part of a much bigger program we call diverse intelligence, which seeks to understand the whole spectrum, all the way from passive matter, all the way up through the different kinds of cybernetic control mechanisms, and ultimately things like second-order human metacognition and so on. Okay, so the next thing I'm going to do is show you some examples of intelligence in the cellular collective. Okay, so what are your cells doing that is actually intelligent? Well, the first thing we know about development is that it's reliable. So a human embryo almost always results in exactly the right species-specific target morphology. But the reliability of it and the complexity of the outcome are not why I call it intelligence. This is really important. The intelligence is not merely complexity. It is not merely doing something reliably. All of that can be done mechanically. That is not what I'm talking about. What I'm talking about is the creative problem solving. And here's an example. If I take this same embryo and cut it in half, or into quarters or eighths or beyond, I don't get half bodies. What I get are perfectly normal monozygotic twins, triplets, and so on. And that's because this system doesn't mechanically just go from its normal starting position to its goal in anatomical space, but it can actually get there from other positions. You can do all kinds of things with this embryo. Of course, it's not limitless. There are things you can do to mess it up. But there are many things you can do where the system recovers. Half of it is missing, no problem. It can tell what's missing, how much, and so on, create the next create the missing pieces and get to where it's going, even avoiding these local maxima in this space. Some animals are able to do it throughout the lifespan. So here's an axolotl, and so this amphibian can regenerate its limbs, its eyes, its jaws, portions of the heart and brain. And when these guys inevitably bite their legs off, as they often do, for each other, then whether that happens down here or whether that happens up here or up here, the cells will build exactly what's needed, and then they stop. That is the most amazing thing about regeneration, is that it knows when to stop. When does it stop? When the correct limb has been completed. That's when it stops. So what you're seeing here is a homeostatic loop. You're seeing a system that it can tell that it's been deviated from the correct position in the anatomical space. It works really hard to get back to where it's going, and then it stops when it's met its goal. Okay, that is what I mean by a goal-seeking system. I don't mean that it's metacognitive and it knows that it has a goal. I just mean that it is competent at pursuing a particular goal, and it will do so even when it's deviated from it. It will try to find its way back. By the way, this kind of regeneration is not just for so-called lower animals. The human liver is highly regenerative. Every year, this animal, deer, it's a large adult mammal, will regenerate huge amounts of bone, vasculature, innervation as it regrows the antlers, up to 1.5 centimeters of new bone per day. It's an amazing regenerative rate. And actually, even human children can regenerate their fingertips. Up until a certain age, they will regenerate them. So we have this problem of regenerating, which shows us that creatures can restore missing parts, but it's actually even much more impressive than that. Here's one of my favorite examples. this is a cross-section through a kidney tubule in a newt. And you'll see normally there's like 8 to 10 cells that form this kind of ring structure around a space in the middle. That's the lumen. Well, one thing you can do is, and this was done back in the 1940s, what you can do is create a situation where there's too many copies of the genetic material at the one cell stage in the egg. When you do this, first of all, you still get a normal newt, so that's kind of wild. It doesn't matter how many copies of your genome you have; you still get a normal animal. The second thing that happens is the cells get very large, so that's interesting. The cells actually adjust size to keep a certain ratio to the size of their nucleus. But there's a problem because if you may have these giant cells and you still try to do 8 to 10, you're going to get an enormous newt. Well, that's not what happens. The newts are normal sized. Well, how can that be? That's because these large cells can create the same structure by using way fewer cells. So now there's maybe only 4 or 5 cells that are making the same lumen. And the most impressive thing is when these cells get truly gigantic. So this is like 6N polyploid newts that have many more copies of their genome. The cells get really big. One cell wraps around itself to create the same structure. Now, a couple of interesting things to note here. One is that this and this are completely different molecular mechanisms. This is cell-to-cell communication. This is cytoskeletal bending. So what you have here is a situation where the goal of making a particular anatomical structure, so that navigation in the space of anatomical shapes towards the correct path, can make use of different molecular components. That's actually kind of the schematic for many types of IQ tests, where they give you a task and they say find creative ways to use the components you already have to solve this task. So in order to do this, the cells literally use a different molecular mechanism. Now, just think of what this means. If you're a newt coming into the world, what can you count on? Well, we know you can't count on your environment, and we know you might get damaged, and so it kind of makes sense that you regenerate. But look, it can't even count on its own parts. You don't know how many copies of your genome you're going to have. You don't know how big your cells are going to be. Your own parts are unreliable, and yet you have to have the goal met. This is what I mean by intelligence. It's literally creative problem solving. It's the ability to handle novel situations by using the tools that you have in new ways. And another example like this is kind of a simpler example that sets up the next thing I'm going to talk about, which is that these tadpoles need to become frogs, and in order to do that, they rearrange their face. So their eyes move forward, their jaws move, everything moves. And you might think that this is a kind of a hardwired process that basically each of these organs just moves in the right direction, the right amount, and then you get a normal frog. So we decided to test that, and this is very critical for estimating the intelligence of anything. You have to do perturbative experiments. You can't just guess. So what we did was we scrambled the position of all these organs, kind of like a Mr. Potato Head doll. We put the eyes on the back of the head. The mouth is off to the side. Everything is kind of shifted. And these animals, it turns out, make quite normal frogs because all of these organs will move in novel paths. It is not hardwired. They will move in novel paths until they get to a correct frog face, and then they stop. And so that tells us that the genetics does not specify a bunch of hardwired rearrangements. It specifies a problem-solving agent. It specifies a system that executes a flexible program that recognizes unexpected starting positions and takes corrective action. So that brings up two questions, two basic questions that turn out to have an answer in common, which is, first of all, what are the mechanisms? You can call them cognitive glue. What are the mechanisms that allow individual cells to work towards large-scale outcomes? I mean, no individual cell knows what a finger is or how many fingers you're supposed to have or where the eye goes. Individual cells don't know this, but the collective absolutely does, because and I've shown you a number of examples where you can see they know exactly where they're going in that anatomical space. So what enables that to happen, and how do they know what the goal is? Right? It's one thing to form a collective that knows more things than its parts, so basic collective intelligence, but how does it know what the correct frog looks like? So this is what we've been working on for some years as a model system. So collective intelligence of groups of cells as a model system for scaling up the competencies of individual subunits into something greater that operates in a new space. This is fundamentally a problem of cognition and collective intelligence. What we have to do is, in addition to this standard scheme that is in all of your developmental biology textbooks, you have gene regulatory networks. They make some kind of protein. Then there's this process of complexity and emergence, and then out comes something like this. Well, we have to add to this these homeostatic loops that I just talked about, which, if there is damage that gets you away from this state, injury, mutation, teratogens, whatever, then this whole system has to kick in and do something different in order to return you to that state. So this unusual way of looking at it, meaning the standard scheme is just kind of open loop, feed forward, emergence. These things happen at the molecular level, and then eventually something happens. But this is actually this error calculation and correction model is something different. And so what it enables is the following implications, which we have been testing for some decades now, which are that, first of all, if you want to change the way the system works, you don't necessarily have to change the hardware. What if you just changed the set point? What if you changed the memory that tells these cells what it is that they're trying to build? Like in your thermostat, if you want it to be at a different temperature, you don't need to change the thermometer. You don't need to change the plant that heats or cools. You can just change the set point. So could we do this? And that means that if we could decode where that goal information was being stored, okay, the memory of this collective intelligence, we could rewrite it and in fact leave the hardware entirely alone. We wouldn't have to change the genes. That's a very convenient feature, because actually this doesn't work backwards. In other words, with a few exceptions, in the general case, if you want to make large-scale system-level changes here, we have no idea how to figure out what we would have to change down here. That is what's kept this inverse problem is what's limited regenerative medicine until now. So it would be nice to be able to avoid that, change the set point, and let the system do what it does best, which is to build to that set point. So in order to think about how to do that, how could we decode what are the memories of a group of cells? We asked, we looked at an example of kind of non-controversial example where cells store memories and act towards them, and that's the nervous system. So we know that in the brain, you have hardware like this. You have cells expressing ion channels. They acquire certain electrical properties. They pass them on or not to their neighbors using these electrical synapses. And that network, okay, so that's the hardware, that network runs a kind of physiological software. Here's an example. You're seeing this group did this amazing video of a zebrafish brain as the fish thinks about whatever it is that fish think about. And that suggests this program of neural decoding. So neuroscientists try to read this electrophysiological information and decode it, because the idea is that the animal's memories, goals, preferences, and everything else are in some way encoded in this electrical activity. So that's the paradigm, that if you have a network like this of subunits communicating electrically, the collective, which is more than the sum of its parts, stores its memories in the dynamics of the electrical signaling. Well, it turns out, and you can see the details in this paper, it turns out that is an ancient system. It's not about brains at all. Actually, evolution discovered this around the time of bacterial biofilms. And so every cell in your body has these ion channels. Most cells have electrical synapses between their neighbors. And now we can say we can run the same kind of program. So neuroscience outside of the brain and nervous system. We can ask, what is it that the body was thinking about before nerve and muscle evolved? These networks have been around for a really long time. What were they thinking about? And so what they were thinking about is shape. They were thinking about anatomical shape. And just as we can try to decode the electrical activity of the brain, we can try to decode the electrical activity of the body to understand how do the cells know what to do in embryogenesis. So we developed some of the first tools to do this, and this is in particular, it started out with voltage-sensitive fluorescent dyes. So we used existing voltage-sensitive dyes to track the outcomes of embryogenesis, of regeneration, of cancer suppression. Here, these are individual cells. This is an early frog embryo, and you can see all of the cells having these electrical communications with each other. Right? And you can see what's happening. There are some fast voltage changes. There are some very slow voltage changes here. So we do a lot of imaging like this. We also do a lot of quantitative simulations. So, for example, we can try to understand how the gene regulatory networks that express channels result in excitable tissues that can form patterns, and how these patterns repair after damage, as a kind of pattern completion in both in neuroscience and in machine learning, we have these architectures where if you're missing part of the input, the system can sort of reconstruct what goes there. So we do a lot of computational modeling like that. Let me show you a few examples of these bioelectric pattern memories. We call this one the electric face. So this is one frame of this video, this time-lapse video of an early frog embryo putting its face together. And there's a lot going on, but what you can see in this frozen frame here is that there's a bioelectric pre-pattern that already tells you what the face is supposed to look like. So here's where the eye is going to go, here's where the mouth is going to go, here are the placode structures out here. You can already see the pre-pattern long before the genes start to turn on to regionalize the face. And this is critical. This bioelectrics allows the cells to cooperate to figure out what the large-scale structure is supposed to look like, meaning how many eyes is it supposed to have and where do the eyes go, for example. Not only is it a cognitive glue binding individual cells into an embryo, it actually binds individual embryos into a group. So here you can see if this guy is poked, these two find out about it in short order. If this one has damage, all the ones around it find out. It's a multi-scale communication mechanism. Here it is on the single cellular level. You can see some neurons, but you can also see a lot of activity out here that is developmental bioelectricity. So this kind of electrical communication to produce wholes out of parts. Now, observing and analyzing these patterns is all well and good, but even more important is being able to change them and to rewrite them. And basically, we took all the tools from neuroscience. We do not use any applied fields, waves, radiation, electromagnetics. There's no frequencies, nothing like that. What we do is manipulate the actual interface, the bioelectrical interface that cells use to control each other. So that means we can control the ion channels. We can open and close them using drugs, optogenetics, and so on. And the same thing for the electrical synapses, for the gap junctions. We can open them and close them. And so that allows us to control basically what in neuroscience would be called the synaptic plasticity and the intrinsic plasticity. So, okay, so those are the tools we have. So molecular biology to control the bioelectric patterns inside of groups of cells. So now can we rewrite the information content of these networks? Can we put false memories into the collective intelligence of cells? So here's a couple of examples. One example is that in an early frog embryo, we target some cells that are going to become gut. We target them with ion channel mRNA, encoding some potassium channels. And what they do is they reproduce that little eye spot that I showed you in the electric face, but they reproduce it somewhere else. So if you put that eye spot, that little voltage spot here in the gut, then what you've told the cells is build an eye here, and that is exactly what they do. These eyes have all the same lens, retina, optic nerve, all the stuff they're supposed to have. And notice a couple of interesting things. First of all, this tells you that the bioelectrics is instructive, meaning that it is actually telling the cells what to do. Second, notice that it's instructive at a very modular high level. We did not tell individual genes to turn on and off. We did not tell individual stem cells what to become. We told a large group of cells be an eye. The signal encodes something a very large scale state. It is not something that individual cells or molecules know. It is something that only the collective knows. And that works precisely because being able to trigger this very complicated cascade of molecular events that are needed to build an eye, being able to trigger them with a very simple signal. You know, all we said is make an eye. We didn't say how big, what the structure of the eye is, and so on. That only works because the whole system as a whole is made up of modules that are good at following specific goals. They just need to know what those goals are. And there's a lot of we could spend a whole hour talking about just this experiment. A lot of interesting things here. For example, if you only inject a few cells like these blue cells, they will actually go out and recruit all this other stuff, which we never touched. Those cells are completely untouched by us, but they will recruit them. It's a kind of secondary induction. Because this system knows how many cells it takes to build a proper eye, and it knows there's not enough of them, and so they will take over the task of recruiting their neighbors. We did not have to worry about that. So you see, when I say it's an agential material, this is what I mean. It has competencies we don't have to worry about. We don't need to know how to turn on which genes to make an eye. We don't need to know how many cells exactly have to be part of an eye. The material does all of this for us, and you don't find this in Legos or pretty much anything else that humans build with, at least to date. The material is incredibly competent at these kinds of things. It's not just about eyes. We can make inner ear like this. We can make ectopic hearts. here. We can make ectopic forebrain like this, ectopic limbs. Here's our six-legged frog here. We can also make fins, which is kind of interesting. Tadpoles don't have fins, and so I'll get into that momentarily, because you can make structures that don't actually belong on that species. Okay, so showing that we can induce various organs in this way by providing bioelectrical states that encode, they have meaning to the surrounding cells. We can use this for a regenerative medicine program. For example, frogs normally do not regenerate their legs as adults. 45 days later, there's nothing. We created a cocktail that triggers a signal that causes the leg to regenerate. So within about 24 hours, you get this pro-regenerative blastema. You then eventually, by 45 days, you've got some toes, you've got a toenail, and eventually a pretty respectable leg that is both touch sensitive and motile. And our record at this point is 24 hours of exposure to the intervention, followed by a year and a half of leg growth, during which time we don't touch it at all. And the reason that is because we are not micromanaging it. This isn't scaffolds, it's not stem cell therapy, it's not 3D printing. We don't touch it at all at that time. The goal is during the first 24 hours to get across the idea that you should be moving towards leg building region in that anatomical space, not the scarring region. And so here I have to do a disclosure. So Morphoceuticals is a company that Dave Kaplan and I spun off, which basically seeks to take that kind of technology into mammals and eventually human patients, right? So there's a bioreactor that we make that is delivering the intervention and providing an environment for regeneration. And then there's the payload, which are ion channel modulators and some other things. Okay. So the next story I want to tell you is to really kind of drill down on this notion of bioelectrics as memory, as pattern memory. So these are planaria. These are flatworms that have a true brain, they have a central nervous system, bilateral symmetry, and so on. And one of the amazing things about planaria is that you can cut them into many pieces. The record, I think, is 276 or something like that. And when you do, each piece regrows a perfect tiny little worm. There's all kinds of interesting problems. Like if you cut them in half, these cells up here have to make a new tail. These cells up here have to make a new head. But they were neighbors right up until the time you cut them. How do they have these different anatomical positions, anatomical identities, rather? So that tells you it's not local decision making. You can't, from the position, because that's identical here, you can't tell what you should be. So how does each piece know how many heads it should have and which end should be a head, which end should be a tail? So we studied this, and it turns out that there's an electrical component to this, which is that if you have a single worm like this, and you cut off the head and you cut off the tail, this middle fragment will reliably give you a one-headed worm. But you can also make the same normal kind of worm, when you cut off the head and the tail, give you a two-headed worm. How do you do this? Well, you do this by noticing that there's actually a bioelectrical gradient that says one head, one tail, and that determines what the fragment is going to do. And if you rewrite that information, and you basically put a false memory into this electrical network that says, well, actually a normal worm should have two heads, then when you do cut them, that's what you get. You get two heads. The cells, they obey the set point and they build the correct thing. Now, notice that this here is not a map of this two-headed worm. This bioelectrical image is a map of this anatomically normal one-headed worm. What we've done is change the voltage, but it's a latent memory. It doesn't do anything until we actually cut the animal. Then you get to find out what the memory was. So the molecular markers, so here's the head marker in the head, not in the tail, and the anatomy are perfectly normal. It's the stored representation of what you should do if you get injured at a future time. That's what's been rewritten here. And so this is a kind of counterfactual memory. It is possibly the basis of what we have in brains, which is this amazing time travel, right? It's this ability to remember things and envision things that are not happening right now. And this electrical network is able to do that. It's able to store a pattern that is not the same as the current anatomy. So the body of the flatworm can store at least two different representations, two different plans for what a correct planarian is supposed to look like. No doubt there's more, but this is what we've nailed down here. Now, I keep calling it a memory because if you take these two-headed worms and you start cutting them in plain water, no more manipulations of any kind, you cut them in plain water, what you'll find is that they continue to generate two-headed animals. Now, notice there's nothing wrong with them genetically. We haven't touched the genome. So the question of what determines how many heads a planaria has is kind of subtle. You can't just say it's the genome, because this two-headed line of animals has a perfectly normal genome. What the genome does is encode the hardware for a system that by default acquires a memory of one-headedness, but it can be changed. Like any good computational or cognitive system, it can be rewritten. It's long-term stable, like any good memory, but it's rewritable. And so, and we can in fact take the two-headed guys and rewrite it back to being one-headed. And so here you can see what these two-headed animals are doing. Okay, so we do a lot of computational modeling to try to understand how do different memories and different representations become encoded in an electrical network. This is, by the way, very parallel to what's going on in computational neuroscience and in AI and machine learning. But now notice that it's not just about head number, it's far more than that. For example, head shape. So here's a planarian, it's a different species with a nice triangular little head. If we amputate the head and we perturb the bioelectrical processes that are normally required for it to remember what its head shape is supposed to be, we can make flat heads like this P. felina. We can make round heads like this S. mediterranea, or of course it can make its own normal head. These animals are about 150 million years separated from these animals, but again, no genetic change here. So the same hardware, that genome made some hardware that is able to navigate regions of that state space, of the anatomical space, and visit attractors that belong to other species. So these other species normally live in these kind of morphological attractors, but this one can visit there perfectly well if it wants to. And it's not just about head shape; it's also the shape of the brain, and the distribution of stem cells becomes just like these other species. So the plasticity is remarkable. You can even go further than this, and you can make shapes that don't look like flat planaria at all. So you can make these crazy spiky forms. You can make these cylindrical things, kind of hybrids like that. The latent space of anatomical possibilities is huge, and the exact same hardware can be pushed into these directions. Now, this is also not this kind of a cool example that comes from the plant world. These are acorns and oak leaves, and you might think that this is exactly what the oak genome knows how to do, is build these flat green things. But along comes a non-human bioengineer, so it's a wasp that has certain signals that it puts out to hijack the morphogenetic capabilities of the plant leaves and cause them to build something like this. Isn't that amazing? Would you ever think that something like this that happens so reliably over and over again all over the world is actually capable? Instead of being this flat green thing, it can actually build these beautiful spiky red structures, and that's because it's been reprogrammed by signals from this non-human bioengineer. In fact, if you look at the different types of structures that are formed, you see kind of a cool relationship. More advanced bioengineers make more elaborate constructions. So bacteria and fungi just basically make blobs like this, overgrowth. Nematodes make some kind of a shape thing, and mites make little round things. But by the time you get to insects, you can make something like this. This is what they do with the agential material of the plant. So again, this is presumably not a self-reflective metacognition. Nevertheless, this is what they're actually doing. They're reprogramming they're taking advantage of the plasticity and the programmability of these cells to do something very different than what the genome normally does. Okay, so our goal is to then have a full computational understanding not only of the molecular biology but the electrophysiology, and ultimately the decision-making, the algorithmic decision-making of the collective, which has to read the electrical information, interpret it, and decide the mapping here that there's a particular state, such as that electric phase, that maps onto a particular anatomical outcome. So that whole process, much like in neuroscience, you can go all the way from the molecules up to the high-level executive decision-making and all of the cognitive processes underneath. And the reason that these models are very helpful is because ultimately what you want to do is make repairs, for example. So Here's an example of taking that approach to deal with birth defects. So here is a normal tadpole brain. You've got your forebrain, midbrain, and hindbrain. If that animal is that embryo is exposed to teratogens, you can see some defects here. So basically, there's profound defects here. And the question is, how do we repair it? And so we created a model. So this is Alexis Pajtak in our center created this computational model where the voltage pattern is what tells the brain what size and shape to be. And so then we were able to see that basically what happens in these teratogen exposed embryos is that the bioelectric pattern is all wrong. And so then the question is, how do you fix it? And the computational model can tell you that if you turn on a particular or off a particular ion channel, you can get back to the shape that you want. And this is what you see here. Again, this is a normal tadpole brain. This is a tadpole. Actually, for teratogens, we used, instead of chemical here, we used a mutation of a gene called Notch. So it's a very important neurogenesis gene. And you can see the forebrain is gone. The midbrain and hindbrain are basically a bubble. These animals are profoundly affected. They have no behavior. They just lay there doing nothing. The computational model told us that we could get back to the correct bioelectrical pattern if we upregulated the activity of an HCN2 ion channel, because it would sharpen that bioelectrical pattern. And so when you do that, you basically get back to a normal embryo with a normal brain, despite the fact that that dominant Notch mutation is still there. Okay, so it has normal anatomy. The animal's learning rates are basically indistinguishable from controls at this point. So you rescue the anatomy and the function with this bioelectrical repair method, even though the mutation is there. So some problems, some fundamentally hardware problems, are actually fixable in software. Okay, so the mutation, and I'm not saying we can do this for all cases. There are many mutations that are not going to be solvable this way, but in some cases you can make up for hardware defects with corrective patterning in software. So where this is going ultimately is that there will be a tool like this where you will the tool will have information about what the correct bioelectric pattern is, and then what the incorrect pattern is in some disease state, for example, tumor, which is what I'm going to talk about next, is cancer. And then there will be a computational component that will ask what channels and pumps could be turned on and off to repair the pattern. And then once you know that, then you can ask, well, what drugs are available to do that? And something like 20% of all drugs are ion channel drugs. So there's this incredible toolkit of electroceuticals just sitting out there to be applied to all these problems with the appropriate computational models. And you can play with an early version of it here. We already have a little bit of this going. So I've shown you an example of doing this for birth defects. I've shown you an example of doing this for regeneration. I want to get to cancer, because cancer is actually a really important component of all of this. So let's remind ourselves of the fact that what happens with this agential material during evolution and during development is that its goals scale. The cognitive light cone of these systems, meaning the size of the goals that they can pursue, really scale up. So individual cells have tiny little goals. The only thing the cell is going to work towards is a specific pH, metabolic, and other levels right around its own surface. It doesn't care about anything else here. But this system has a huge, grandiose goal. It knows it needs to have 4 fingers and be a certain size. No individual cell knows this, but the collective absolutely does. Because if you cut off fingers or you cut off the whole thing, it will do exactly what it needs to do to get its goal met. So the collective has a very large set point. These guys have little, very tiny set points. And I've just shown you that the way the cognitive glue that binds individual cells towards systems that can store and pursue large goals is bioelectricity. It's the electrical networks that allow these cells to remember what a limb is. That has a failure mode. That whole cognitive glue system has a failure mode, and that's called cancer. What happens in cancer is that cells physically disconnect. They electrically disconnect from their neighbors, and as soon as they've disconnected from the electrical network, they can no longer remember any of these large-scale goals. They just go back to their ancient, primitive, tiny little goals. And that is go wherever life is good and proliferate and so on. And that's metastasis. So that's what you're seeing here is individual cells, which are glioblastoma, basically rolling back to their unicellular past. So that's what we have here. We have individual goals tying together via bioelectrics and other signals, of course, too, biochemical and biomechanical signals, into systems with a much larger degree of both temporal and spatial goal directedness. And that suggests that very weird view of cancer and cancer suppression suggests an alternative medical modality. So, for example, what we could do instead of trying to kill these rogue cells, what if we tried to reconnect them to the electrical network, despite whatever problems they had? What if we try to reconnect them? We already know from both the brain repair case and from the two-headed planaria that genetics does not tell the whole story. That you can actually what actually drives is the physiology and that's operating in these cells. So what if we tried this for cancer? So here it is. We inject a human oncogene, KRAS mutations, P53, things like that. They will eventually make a tumor. But if you watch that spot, you can already tell where the tumor is going to be. This is again a voltage dye map. You can already tell that there's some aberrant voltage going on here, and these are the cells that are going to disconnect and basically treat the rest of the body as just external environment. Okay, they don't consider themselves part of the whole anymore. It's just outside to them. They're going to go wherever they want, do whatever they want. Well, if we co-inject with that oncogene an ion channel, that forces these cells to have a particular voltage pattern and to stay connected to their neighbors, then this happens. While this is the same animal, while the oncoprotein is still blazingly expressed. Okay, so there's mutation all over the place here. And if you sequence it, you would make the wrong prediction. You would say that, well, yeah, there's definitely going to be a tumor here. Look at how much of this oncoprotein there is. But there is no tumor because that's not what drives. What drives is not the genetics. What drives is the physiology. That once you've connected the cell to that neighbor to their neighbors, the collective is saying we are building nice skin, nice muscle, and so on. The collective does not have thoughts about migrating away and doing something else. What it knows how to do is navigate anatomical space to keep this tissue healthy. So. This is both a diagnostic and a therapeutic modality that we're pursuing now in human tissues for cancer via this kind of boundary of the self approach, where we try to inflate that boundary that shrinks, the boundary between self and world that shrinks. So the idea is that these cells are not anymore these cancer cells they're not any more selfish than normal cells. They just have smaller selves. Okay, all right. So now for the last little bit of this, I want to shift from. So far we've had a focus on native outcomes. In other words, let's repair the birth defects to the correct shape. Let's regenerate organs that were there before. Let's prevent or normalize cancer so that you can have normal tissues. Now we're going to go beyond this. Here's the bioengineering part of this talk. We're going to talk about biorobotics and specifically this idea of novel anatomical goals. So I've shown you systems that pursue evolved anatomical goals, such as having a proper face, the proper number of heads, the proper structure of the brain, and so on. But what about novel systems? Where do they get their goals, and what are their goals? And famously, Feynman wrote that what I cannot create, I do not understand. So the idea is by synthetically creating novel beings that have never been here before through evolution, we gain some understanding of where goals come from in the first place. And so this is work largely spearheaded by Doug Blackiston in my group, and our collaboration with Josh Bongard's lab in our Institute for Computationally Designed Organisms, where we wanted to know, could we use cells liberated from their normal context, the embryo? Could they reboot multicellularity? What would they want to build? And so what we did here was, here's an early frog embryo. Here is the cross section. These animal cap cells normally make ectoderm, so kind of think perspective skin layer. So we take these cells, we dissociate them when we put them in a petri dish. Now, what could they do? Well, once you do this, they could do many things. They could die, they could crawl off and get away from each other, they could make a two-dimensional monolayer like cell culture. But instead what they do is, overnight, they compact and they become this little thing. The flashes you're seeing is calcium reporter, calcium signaling. And they become something we call a xenobot. Well, the first thing that happens actually is we call these microbots. Each one of these is an individual cell. And this little thing that remarkably looks like a little horse is just one of many examples where you can see they sort of get together. They have these interesting movements. They kind of start interacting with each other. There's that calcium flash. You can see how this thing is sort of skipping along here in the petri dish. And there's all kinds of little structures that you can see, and eventually they all come together and they make this. This is our xenobot. We call it a xenobot because Xenopus laevis is the name of the frog, and we think it's a biorobotics platform, among other things. So xenobot. So what they're doing is they're swimming along because they have little hairs on their surface that row against the water. So these are cilia that frogs normally use to push mucus around on their skin. They can go in circles. They can kind of patrol back and forth like this. They can have group or collective behaviors. This is some tracking data. You can see this one's going on kind of a long journey. These two are interacting with each other. Some of them are kind of hanging out doing nothing. We can make them into weird shapes like these donuts that then swim around. Here's one traversing a maze. So there's no fluid flow. It swims down here by itself. Takes the corner without bumping into the opposite wall, and then for some reason decides to turn around and come back. So these are completely autonomous. We're not pacing them. We're not making them move. So it swims here, takes the corner without having to bump into anything, and then kind of turns around. They also have the ability to self-repair. So if you chop them almost in half, they will fold back up like this. Look at it's like doing a bicep curl from 180 degrees. Right? Like that hinge is generating a ton of force to fold this thing back up. They do have these interesting flashes. You know, this calcium signaling is how people profile activity in the brain. And so one thing you might ask, and we have a preprint of this, even though there's no neurons here, you might ask how similar is this kind of calcium activity? What's the computation that might be done here? How similar are they to brains and so on? Even though this is just skin. And one of the most remarkable things they do is something called kinematic replication. Now, we've made it impossible for these guys to reproduce in the normal froggy fashion, but if you give them a bunch of loose skin cells, what you find is that they gather these cells up into little balls, and they polish them, both collectively and individually, and these little balls mature into the next generation of xenobots. And guess what they do? They make the next generation and the next generation. So it's a kind of kinematic self-replication, and it works because evolution, just like us, is working in an agential material. These cells are not passive particles. They are cells themselves that will actually become xenobots. So the amazing thing about this, one amazing thing about this is that there haven't been any xenobots before. There's never been any selection to be a good xenobot. Where do their properties come from? You know, what did the frog genome learn during its many years on Earth? Well, it certainly learned to do this. So it's hardware that can make this set of stages with behavior. You know, here's some tadpoles running around. But apparently, that's not the only thing it can do. It can do this. And while this was under strong selection, evolutionary selection for specific environments, these never directly existed until we made them. So this is an 83-day-old xenobot. You know, what is it becoming? What shape is this? Where does the kinematic replication come from? It's a really remarkable thing to be thinking about the goals and competencies of creatures that have never been here. So you can't say that it's evolution that gave it to them. They have no straightforward evolutionary backstory like all the other animals that everybody studies. Soon we will have out some actually behavioral studies. So stay tuned for that. We've been studying their ability to sense, to remember, to behave, and so on. And that's all coming. Now, one thing that you might think of is that this is some weird, unique fluke of amphibian and embryonic development. I mean, amphibia are pretty plastic. Embryos are definitely plastic. Maybe this is just some kind of frog thing. You know, maybe this is just applicable to frog embryos. So I'm going to show you this. If you look at this thing, and I ask you, what do you think this is? You might think that this is we got this out of the bottom of a pond somewhere. You know, some kind of primitive organism. And if I ask you what the genome would look like, you might guess that it would look like the genome of primitive organisms. If you did sequence it, you would find that it's 100% Homo sapiens. These are 100% human cells. They are not embryonic cells. They are taken from the tracheal epithelium of adult human donors. And much like the xenobots, when given the opportunity, these cells will take that chance to have another life. They reboot themselves into a novel creature that doesn't look like any stage of human development, and they have a completely different structure and some very different properties. And again, they are self-motile. One thing that they can do, for example, is traverse wounds. So this is a big scratch wound placed in some human neurons. So here, this is a lawn of human neurons. There's a scratch that we made, and they can traverse it. When they do traverse it, they can sort of collect in a pile. We call this a super bot cluster. It's probably around 10 bots or something like that here. And what you can see, what they start to do, and you can see this clearly when you lift them up 4 days later, what they're doing is they're sitting there knitting the two sides of the gap together. Okay, so now just think, who would have thought that your tracheal epithelial cells that sit there quietly in your airway doing nothing other than moving mucus and dust particles and so on, have the ability to reassemble into a completely novel little organism with a completely novel transcriptome. I didn't show you this, but they actually transcribe their genes. About half of their genome, half the human genome is transcribed completely differently in these guys, just because of their new lifestyle. There are no transgenes. There is no synthetic circuits. There's no synbio here. No scaffolds. No weird drugs. We didn't edit the genome at all. This is completely normal cells doing something that nobody had expected them to do, and they have the ability to exert repair. So we think this is actually the beginnings of a technology for personalized autonomous therapeutics. We could take cells out of your airway, use them to create anthrobots, and put them back for a range of applications, which we're just now scratching the surface of. For example, neural repair, but in a way that does not require immune suppression. Yeah, it doesn't. They're your own cells, so you don't have to worry about protecting them from the immune system. You wouldn't be worried about any kind of nanomaterials or anything like that accumulating in the body. They're completely natural, made of your own cells, and they degrade after a few weeks. So we think this is the beginnings of these kind of autonomous therapeutics, which, by the way, are way smarter than anything we can build today. They have 1 billion years of living inside a body. They already know what inflammation is, what cancer is, what stress is like, and so on. So I think the sky is kind of the limit in learning to use these things for therapies. So just to wrap up here, just a couple of things. First of all, I want to point out that the whole field of biomedical interventions can be kind of categorized as bottom-up or top-down. This is everything that's been going on until now. So surgery, stem cells, genomic editing, synbio, and targeting drugs with molecular targeting receptors with molecular drugs is all kind of bottom-up, hardware-focused approaches. The future of biomedicine, I think, is complementing all of that with really powerful top-down approaches. So behavior shaping, I didn't even talk about all the cell and tissue training that we're doing, agential implants such as anthrobots, and all of the morphoceutical and in particular electroceutical signals that are available for getting cells to do very complicated things that we do not want to micromanage. The name of the game here is going to be cooperation and collaboration with the material, not micromanaging. And we actually think that AI is going to be a really important component of this, to be able to crack the communication interfaces so that we know how to talk to all the different levels. Okay, the way we talk to each other right now at the highest cognitive level, we should be able to talk to all of the somatic intelligences within the body. And I think that future medicine is going to look a lot more like kind of somatic psychiatry than it's going to look like chemistry. Okay, it's going to be quite different. So that's for biomedicine. Now for the bioengineering and the biorobotics is, again, in many cases, you don't want to build from scratch where you have to micromanage all the hardware. The material that we're dealing with already has built-in sensors, amplification machinery, the ability to discriminate signals, to generalize, to learn, to adapt, to make decisions, and so on. I showed you many examples. And so taking advantage of this is the frontier of biorobotics, is not trying to force specific molecular states, but actually communicating and collaborating and reprogramming these existing capabilities. Basically, the AI discovered bioprompting of the material. Now, the very last thing I want to point out is to kind of step beyond medicine and engineering to the kind of ethics component of all this, which is the following. this is what I call the spectrum of persuadability. All systems are somewhere along a spectrum like this, related to what kind of approach you're going to take to relate to them. So very simple machines which are only amenable to hardware rewiring. And then you got these cybernetic things like thermostats and so on, where there's a set point and you can modify them by controlling the set point. Then there are some systems that learn, and you can use behavior shaping and rewards and punishments and so on. And I've argued that cells are somewhere in this neighborhood. It's not just brainy animals, but actually cells and tissues are in this neighborhood too. But notice what happens, and then of course all the way to the right are very complex high-level intelligences with which you communicate by reasons, not even rewards and punishments necessarily. But notice what happens as you move to the right of the spectrum. Here it's all about control. Everything you're doing is exerting control over the system. But as you move right, it's less and less exerting control and more communication and collaboration. And by the time you get to something that's up here, it's actually a very bi-directional relationship, because the level of agency rises here. And by the time you're having this kind of interaction, which is not about control or manipulation, it's about bi-directional, mutually beneficial interactions. It's about communication and thriving together as high agency systems. So the reason I bring this up is because of life's incredible interoperability and the future of bioengineering. We and certainly you as students in this audience, in your lifetime, absolutely are going to be living with a very wide range of beings. Okay, everything that Darwin meant when he said the natural world and its endless forms most beautiful is like a tiny little region in this whole space of bodies and minds, because every combination of evolved material, engineered material, and software is some kind of possible embodied being. Hybrids and cyborgs, augmented humans, and weird combinations of material extended both biologically and technologically. All of these things we are going to live with, and most of them are going to be nowhere on the tree of life with us. Okay, they're quite different, and this means that we're going to have to develop novel forms of ethics that go beyond what do you look like and how did you get here, meaning evolved or engineered, in terms of knowing how we deal with others that are not quite like us, so that achieving some sort of ethical synthbiosis with a very diverse range of beings goes way beyond the kind of control goals that we have in engineering, and more like a mutual kind of bi-directional mutual vulnerability and relationship. So, okay. I'm going to stop here. I'm already way out of time, and just say that lots of this is discussed in detail in these space papers, both on engineering and medicine. Really critical. I want to thank all the people who did the work. So here are the amazing postdocs and PhD students who did all of this work. We have lots of incredible collaborators, funders, our several companies that support us, so I have to mention those, and most of all the model systems. Okay, the animals that teach us everything we know, because they do all the heavy lifting. So I will stop here and take questions.